Wonderful to be here. It's a huge honour. I've always enjoyed coming to Wellington since I was a young fella, and so it's really nice to be back. I had commented that I don't think it's always been weather like this when I've come, but people have disputed that. But from that very upbeat intro, I'm going to tell you about some macabre things. So we work on dead proteins, so they zombie proteins, if you will. So they look like they should be active, but because they lack key catalytic residues, they're dead. And one of them in particular that I've focused most of my career on is a protein that kills cells. So it's not only dead, but it kills cells. So hence the very miserable imagery. So when I came into this area about 20 years ago, we were already interested in these enzymes called kinases. So they are really integral parts of all of our cells and throughout life. They convey signals. They put a phosphate group on a substrate and they can control all manner of functions of proteins inside cells. And so I've summarized these here, but I guess the big question at the time was, well, what if the protein doesn't have any catalytic activity? And that's really where we've focused. And it turns out that the proteins without catalytic activity can also perform most of these functions. And so that's something that we've delved into, really interested in how they interact with other proteins to regulate their functions and convey signals inside each and every one of our cells. We've had a bit of fun with this over the years, so zombies in particular have featured quite heavily, and we've lifted hard from George A. Romero's back catalogue. But it also dawned on me when I gave a presentation once and in the US, and a colleague there who is from Timaru originally pointed out my comment about these pseudokinases being the black sheep actually has some relevance to New Zealand. And so I'm dying to write a review with this as the title, which will happen in due course, I imagine. We're already planning one called Zombieland, so stay tuned for that. But most of my career is really focused on a protein called MLKL. So when we started working on it, I'm now in my 20th year of working on this protein. And when we started working on it, we didn't know what it did. And it was only 2012 when we learnt of its function in cell death. So we've worked on it at every level imaginable, from the atomic, the cellular, and also using mouse models, as well as looking at what happens in patients when the pathway goes awry. And so MLKL kills cells with this lytic mechanism. So it's this pathway called necroptosis or programmed necrosis. So the famous cell death pathway would be apoptosis, because we've been studying that for the order of 40 years. And so this is a good clean exit. So, and really important developmentally, it's one of the reasons most of us don't have webbed fingers and toes. So it's designed to remove spent cells or in those developmental processes without eliciting an immune reaction. Necroptosis, which is driven by this protein MLKL, on the other hand, is designed to swell the cells until they explode and release their contents and tell ourselves that there's an immune reaction required. So it's designed to make a mess, and that's the reason for why it's lytic. And we've really been focused on trying to understand what's happening at every level, from the organism through to the atomic. And one of the things we're able to do using structural biology, so these are two crystal structures that have been morphed between. This is the open form, but we've also solved structures which show a closed form of MLKL. And so the open form is the unactivated version. The closed form is the activated form, which is the one that can permeabilize membranes and kill cells. Now, in terms of disease relevance, the pathway itself has come about as an innate immunity pathway. But what's attracted a lot of interest is when the pathway goes wrong. And so a number of diseases have been implicated as having errant necroptosis at the heart of them. I'm not convinced that this list is entirely accurate, but we've focused and also demystified some of the role of necroptosis in this. And in particular, we've focused on barrier tissues like the gut. And we've had a lot of interest in IBD, inflammatory bowel disease. And I'm just going to give you a vignette of a study which we're hoping will be formally published soon, also to highlight the contributions of some key New Zealanders highlighted here in this work. What we've done is a clinical cohort study where we've taken biopsies from patients with inflammatory bowel disease, so both Crohn's and colitis, And what we've done is analyze these at every molecular level imaginable. So at the protein level, the transcript level, and also using spatial transcriptomics. And that's the work led by my colleague Edwin Hawkins, who is originally an Aucklander. And so what we've found from this is that in IBD, we see cell death is running amok, but not how we thought it would be. So in the tissue that looks normal, we see necroptotic signaling operating, whereas the lesions, the most inflamed lesions, have apoptosis dominating. And that's the opposite to what we thought we'd see, which is a really cool finding. And it seems instead that necroptotic signaling is smouldering away in these tissues, and we think it's the first domino to fall before we see these lesions forming. And the process seems to be driven by two important inflammatory cytokines, interferon gamma and TNF. And we think on this basis that we can target the necroptosis signaling pathway, and MLKL in particular, as a novel therapy for inflammatory bowel disease. So this is really the culmination of, as I said, more than 19 years work. And we figured from this that, you know, it's cool working on one protein, but what if we could take everything that we've learned, and particularly the cul-de-sacs we might avoid, to be able to expedite understanding other proteins of unknown function. And so we are aware of a number of, also keeping with the macabre theme, the dark kinome. So this is understudied proteins, or about a third of kinases are thought to be, they are undercharacterized according to an NIH classification that happened. And so we thought, hey, why don't we take what we've learned from studying MLKL for a long time and apply the same approach as a platform to understanding some of these proteins. So I'm not going to drill too far into this, but one that attracted some interest for us was a protein called PSKH1, which is a woefully understudied kinase, but it turns out it's really important. So this is an unpublished finding here, but if you take the PSKH1 gene away in prostate cancer cells, you restrict proliferation. So we think it's a really interesting new prostate cancer target. And we weren't the only ones. So here's Chris, originally from Horswell in Christchurch. The video's not going to play, but he's with John, who's on the right, our long-term colleague at MIPS, Monash Institute of Pharmaceutical Sciences. And so what we found collectively was the molecular mechanism by which PSKH1 can be regulated, and gives us some clues as to how it might be inhibited in disease. So obviously, PSKH1 is just one example of what we might learn in future. So there are plenty of things to keep me busy for the next few years. So there are more than 100 kinases and pseudokinases that are understudied that hold our interest, mostly because they might be drug targets in a number of different disorders and diseases. And really, just to finish, it's really fun making discoveries that keeps me going. But I think the real highlight has been being able to work with amazing people over the past, well, more than 20 years. But some of them are shown here. So this is a reasonably recent lab photo. We couldn't get everyone together. Just to point out, I'll tell you why in a moment. Sam on the left has been a key figure. He's worked with me since before I had my own independent research group, so the order of 17 years. So he's an important figure. And my old PhD supervisor is over there on the right, Ian Young. And so he's now part of my lab. He's retired, but he comes to lab meetings. So, and you might notice a bit of a family resemblance between Ian and Sam as well. So they're kind of like extended family to me. And just to wrap up, there's a whole bunch of people who have helped. This is a long-standing project and multidisciplinary. We've had a number of key collaborators who are highlighted in bold here, but also quite a New Zealand connection, both within our institute and locally, as well as a number of people who are based in New Zealand who I've collaborated with over the years. And the last thing I wanted to mention was how important friends and family have been as an amazing source of inspiration and support. My parents are still in Christchurch. I know they're incredibly proud of me, and also my family in Melbourne. So my partner Miranda and our kid Fred, they're very patient with me, whether it's doing an all-nighter at the synchrotron or whether it's going away to travel to meet colleagues or present our work. So thank you very much.